study biology. And there is another small part of the story is about that we humans and other mammals and other animals that I really don't have much clue about them. My original training is pediatrician. I still like mammals as humans more than anything else. We live together and we have a lot of microbes. So for those who go to sleep now, just remember this and it's good enough. Please don't snore loud. Okay. Having said this, I want to start with something. So about 15 years ago, something like this maybe, what happened with biology? The biology became technological science. Basically, biology took advantage of technology that suddenly we became able to measure a lot of things at the same time. Suddenly we could measure thousands and thousands of proteins, genes, metabolites, and so on and so far. And it made the question became for biologists who were basically focusing their attention and brain on what specific thing I'm going to do because I cannot do too many. Now they refocus their brain into how I deal with all these wonderful data that I can get just studying some disease or something else. And what basically usually people do, and they're doing it still now, and there is nothing wrong in doing this, is basically they measure all of this and say they compare. Everybody here heard here about t-tests, right? You compare two states, this is normal, see what is different, and then we believe that there is something behind there which is important for the understanding how disease happens. And this is kind of partial true. There are details behind, but there is nothing wrong with the idea that what is changing is there is underlying cause and process. And the problem with this story is that it's actually very difficult to understand which one of those guys, and here usually each column is a sample or mouse of human, and each line are many, many different genes, metabolites, and proteins. Which one of those is important? Which one of those is critical? And how they make up actually the actual process, physiological process together. And for this, right now what we are using networks. And these networks we are using as a models, and we use those models in order to not just look at this car, but we also want to drive this car. So what, because we believe that understanding, it means that I know where, how I can drive this car. And this is what we are going to do here. And what I am going to tell today, just for those who will sleep, I'm going to explain you how network helped me answer the questions that biology is answering. Different type of questions. It's nothing like mathematics, it's nothing computational. It's actually about biology that is helped by informatics. Oh, sorry. So do you hear me? It's fine? Okay. So here we go. So what we start with measuring a lot of different things, and then we look what is different, and then we construct networks. And the most simple way to construct network or find connections between those variables, those genes, is one of the simplest ways calculating correlations. And now we have a network. The network is a statistical model of the process that we study, how something normal becomes disease or opposite. So what we do next is we investigate this network in order to find subnetworks that are responsible for specific particular process and how they interact with each other and which actually processes are involved here. And once we have the answers of the analysis of the network, what we do, we go back and we test our predictions about specific genes or specific pathways that we predicted to be involved. Once we tested those, we can go back and study further. And basically what I wanted to say is that most of the things that we do, we use something ready and we don't develop many algorithms, but we have to do it sometimes. And we collaborate with mathematicians, and we produce new type of software sometimes, but it's only in case there is a gap in knowledge. So our major objective is not developing new analysis, which we do, but rather answer biological questions. And I wanted to talk today about my research program. So my research program, as we discussed with Oleg, is big, and don't worry, I'm not going to talk about everything that I do, but I just needed to introduce. So the core of this research program is host microbiota interactions. Any of those things here are somehow involved interaction between host and microbiota, and I will tell you later what is microbiota. And we use, for most of the things that we do, network analysis as our key tool to discover biology. So the first story I start with is probably the story that was, I guess, one of the ones that is probably most simple because it doesn't involve that much of microbiota yet, and it's about cervical cancer. And just to mention, I don't have space to talk about my collaborators later on. It's too many of those. So this work is done in collaboration with 2 different countries besides United States and a couple of labs in United States, Norway, Brazil. And what we were doing in cervical cancer is a question about how cervical cancer works. So let's start with cervical cancer just to understand what it is. Cervical cancer is a virus-mediated disease, virus-caused disease. All of you probably heard about human papillomavirus. Does anybody here didn't hear ever about human papillomavirus? Okay, so all of you know that human papillomavirus is a cause of disease. However, most of you probably never heard about that it is actually self-treatable infection. Most of women would eliminate this virus, and only very few actually end up with invasive cancer in case when you then don't go to the doctor during the 10, 15 years of, or 20 years sometimes, of this chronic infection. And very small proportion of those women, as I said, very small proportion is not eliminated virus right away within the first 3-6 months after infection. So what for you to remember at this point now, which is very important to understand what we study, is that it's a complicated story. The episomic virus or wild virus is infecting, and then at the end it's eliminated. However, there is an integrated virus. So episomic virus is cut, and it integrates into the human genome. And at the end, when you have invasive cancer, most of cancers, and this is a rough estimation, it should be 100% probably, invasive cancers have integrated virus. Why is this? So we go to biology now. The biology is complicated, is usually, however, we have known some things about this. There are 3 major proteins of this virus, and this circle represents actually wild virus, which is E6, E7, and E2. E6, E7 are major oncogenes of human papillomavirus. And E2 is a gene that actually blocks activity of those 2. Interestingly enough, when the virus is integrated, it's cut in E2, so E2 doesn't work anymore. And then it's integrated in the human genome, and then what happens is E6, E7 is highly expressed, and the oncogenes works, and cancer happens. What is not clear, though, it was not clear at least before our work, why woman who was not able to eliminate for 10 years, 15 years, this wild virus, that she has some problem with her immunity, suddenly becomes able to eliminate this virus. So this wild virus goes away, and E2 doesn't block anymore oncogenes. So how we study this? So this is a question that is basically was the most critical at the time we were studying this. And what we've done study with these big data. And there is two types of big data here. You can see one is a meta-analysis of gene expression. A lot of red guys are basically upregulated genes, and green are downregulated genes, about 1,100 gene differentially expressed genes in this cancer. And here you see the chromosomal aberration. Chromosomal aberration is another type of data. It's genomic data. And red big pigs are frequent chromosomal gains. So you have more copies of certain part of the DNA, of certain piece of the chromosome in majority of women who have this cancer. And green guys are those that are losses. So what we do here, we reconstruct the network and integrate two types of data. And what we found very interesting result, which was partially surprising, partially not, retrospectively speaking. We found the gene regulatory network that consists of cell cycle upregulated genes, which is not surprising for cancer, epithelial cell differentiation, downregulation, and upregulated antiviral response. And this was not shown before us, and probably we could find it only because we were using networks. What was important here is if you look at those networks, and this is a cell cycle network, and this is antiviral network, we could identify causes of regulation in aberrations. So in the gains, you have causes, and among hundreds of genes within a big piece or big chunk of amplified DNA, you have few genes that are actually driving the whole process for both subnetworks. So we could not find for epithelial differentiation, but we find for cell cycle and antiviral. So in this analysis, this is all computational analysis, and we are so sure about our predictions, but it's not sufficient probably for normal biology. So what I was telling you, we are going back and we are going to test what we predict. So our prediction is this particular gene, and we were very much interested in this particular gene because this was totally novel gene for regulation of interferon antiviral response. We were asking the question whether this gene is actually a regulator or not. And first we looked in the tumors, just inside tumors, expression of genes that are connected to this LAMP3 and regulated by LAMP3, upregulated by LAMP3, in the gains of LAMP3 or no gains. And we look at the ratio gains or no gain, and you see most of genes that are predicted to be upregulated is actually high expression in gains. So it seems that it's actually doing what we expect. But we need to another test, and we do this another test, and we do a siRNA experiment where we block LAMP3 and see what happened with genes. And we actually see that as we predicted for majority of genes, besides one actually gene, our prediction holds true, and majority of genes are actually regulated by LAMP3 in the way how we predicted it's supposed to be. So very important test now. Cell cycle drivers, and this is not published data. The paper is published already, and now we have a new wonderful graduate student in my group, and Daria Vishenska over there, and she is doing the test of our published predictions. And look, out of our 6 top predictions, we have inhibition of 5 leading to defect in cell growth. And when we test for the same thing antiviral driver, it doesn't do it this way. So it looks pretty reasonable for me as a validation of what we are studying for the first result and for the second one. And the conclusion, the big conclusion for cervical cancer here is gene expression program of cervical cancer is induced by driver genes located in chromosomal aberrations. And this is how we prove this. So what is important about disease now? Now we understand the disease. Now we understand why women would be eliminated the virus. What happened is that actually the antiviral response is located in the chromosomal aberration. So only clones that have chromosomal aberrations and actually chromosomal drivers that are other data. I don't have time to talk about this here. So they're sitting together in one chromosomal aberration, and one part would be driving antiviral response, and another directly cell cycle, and this is how cancer would progress. So unless you have, the aberration is random event. So those who wouldn't have those aberrations, they wouldn't progress. And another thing is, it's not woman. It's actually tumor itself is driving antiviral response that is required to get E6, E7 escape from the control of E2. So this was very interesting thing to solve the biology problem, but it also has other important implications. What are the implications of this study? So we're explaining why antiviral therapies may not work in all tumors. Why they may not work is because if you have a lot of integrated virus, you eliminate wild virus, you actually liberate a 67 and you have more oncogene working. But if you early enough come in with antiviral therapy, this will be extremely beneficial. So this is why we have controversial results with antiviral studies. Another thing is early enough detecting expression of those drivers would indicate the progression of cancer. There is another topic that we work actually in collaboration with Dr. Taratula, which is personalized therapy, and hopefully with some funding eventually coming to us, we can proceed. It's a very exciting area because we can block those key drivers of cancer that are present in individual tumor of individual patient. We block them in combination, and it can be very beneficial. And there is another new direction of these studies is, as you know, virus infection can depend on other ones. And role of microbiota in cervical cancer was not really studied, so this is what we are also studying in our group. And I want to talk a little bit about microbiota. So for those who never heard this word, microbiota is basically a lot of different microbes that live with us. And as far as we know now, we have as many bacterial cells as human cells in our body. And we have whatever 50 times more bacterial genes than our own genes. So this is very important organ, our organ, or it's part of us. And actually this is why we probably believe this is important. So if you look in our body, in each place of our body that we contact with environment, we have some different type of bacteria living in us, and they're very, very different depending where we look at. Not surprisingly, during last maybe 5-10 years, I guess 10 years, yes, the first paper is 2006, there are multiple papers showing the effect of different normal microbes living on us and with us on development or protection against different diseases. For example, the first paper I'm showing here is about role of microbiota in obesity. The most recent paper is about role of microbiota in the efficiency of immunotherapy against cancer. This also different, and there are papers now showing the effect on depression and brain development, autism, and so on and so forth. So microbiota is a very interesting topic. And when we decided to study this, and it was actually done in collaboration with our Norwegian collaborators, and study was done mostly at the beginning by our undergraduate student, Kim. So we found that, first of all, very important, we could find bacteria inside of cervical cancer, inside of the tissue. It's not superficial, in tissue. Second, if you look, the cervical cancer is green, and if you look, actually, if you look compared to all different type of microbes, what you see is very close to vagina. When we compare it to vagina, the frequency of microbes is totally different. But it's the same microbes that are living in vagina are moving in cervical cancer. And what we decided to do, we decided to use networks to investigate this. So, and how we do it? So we integrate, like we integrated before, chromosomal aberrations with gene expression. Now we are integrating microbial data with mammalian data. In this case, it's human, woman.
And what we do, we basically make one network for microbes and another network for human, and we integrate it. And we use this network to investigate which microbes can be contributing. And this is another view on the same network I showed to you with cell cycle, antivirus, monoepithelial cell differentiation. And here we have several different particular frequent microbes that connected to one or another subnetwork. We are testing now in collaboration with Alexandra, actually, and Richard, the possibility that those microbes can be playing a role in regulating the critical genes for this cancer. So for cervical cancer, I'm done. And if anybody wants to ask something, I'm happy to answer, or we can delay it afterwards. Anybody wants to know anything about cervical cancer? No? Okay. So the second story. So you probably noticed at the beginning this weird abbreviation, CVID. So CVID stands for common variable immunodeficiency. And common variable immunodeficiency, why we study this, is it's something that was actually our first study that we started study microbiota. And we didn't think about microbiota back then. The microbiota was answer for us. And what is interesting in this case, so we've been studying the mouse model and study a little bit of people. And now we actually study the human disease in collaboration with our colleagues at NIH. And I want to tell you a little bit about common variable immunodeficiency. Although it's extremely rare disease, 1 out of 25,000 itself, it's the most common immunodeficiency, primary immunodeficiency. Second, some of those patients, and we know this now, and we didn't know it before because they were not treated for other problems before, but now some of those patients develop chronic enteropathy. Major problem with this chronic enteropathy is not able to absorb fat, and basically they have chronic diarrhea, they have fat in stool, and they actually have problem opposite to obesity. I prefer to have this problem a little bit. So anyway, for these patients, unfortunately, we don't have treatment. So they don't respond to treatment, and this is why we need to study them, because if we understand how disease work, then maybe we can find the treatment for those. So here is our results mostly of our previous study, and why it is important is because I want to claim that conclusions that we make from mouse research, we can somewhat extrapolate to human disease. So if you look in the most of clinical symptoms, and on cellular level, and on molecular level, they're extremely similar. It's surprisingly similar what we see in human and in mouse. However, mouse we can manipulate, meaning mouse we can do a lot of different things. In particular, we make mice germ-free, and by making those mice germ-free, B-cell knockout mouse, you cannot see what is here, but this is B-cell knockout mice. So B-cell knockout mice is B lymphocyte knockout mice. They also don't have antibodies. It's the same with common variable immunodeficiency. They don't have antibodies. So these mice, when you make them germ-free, the problem disappears. So we have proof. Do you see it here? You have proof that microbiota plays critical role in this syndrome in mice. And we extrapolate based on all other evidence that it's extrapolatable, it's generalizable, that we potentially humans, we have the same problem. Why is it important? It's important because previous studies from very good, high-quality scientific groups were looking desperately to find pathogens as a cause of this enteropathy, as they didn't find it anymore. At the beginning, it was something about Giardia, but not Giardia anymore, no any known pathogen, and they still don't find anything. There are recent reports about some viruses, but basically until recently it's nothing conclusive. This is why if we know that microbiota is important and know the known pathogen, we need to look further. Okay, so CVD enteropathy, we now study humans, not mice, and I want to discuss with you 3 very important questions related to this disease that we actually were lucky to approach and get some conclusive results afterwards. And first question is why some patients with CVAD develop enteropathy and others not? So if you notice on my previous slide, it was 70 out of 300, so not all of them do it. What is the actual reason? And the answer to this question is interesting because I didn't need the network analysis to answer this question. All other things that I'm going to show you, we need definitely network analysis to answer the question. So we'll look at something that is called antibody and called IgA. And IgA is a major antibody in the gut and in general on mucosal surfaces. But in the gut, this is the most important antibody. And we didn't look at intentionally there. We actually did something that I'll tell you just very soon. And if you look at those patients that GI CVAD stands GI symptoms or gastrointestinal symptoms CVAD or no GI CVAD. So basically this is people without and those people with disease, with enteropathy, and they have no difference whatsoever in the serum level of IgA. Agreed. In the paper, several papers showed this, there is no difference. What we found after doing RNA-seq and biopsies of those patients, very surprising result. Look here, IgA is present and very detectable, at least on gene expression level, in CVAD patients that are not supposed to have any antibody, but we see the actual level of transcript, and somehow in CVAD patients that have enteropathy have no IgA. We also confirmed it by looking at the protein levels, and as you can see together with me, and this is a quantitative graph, and this is representative images, yes, here is level is lower, but it's definitely detectable, and here we literally have nothing. And we do believe that we now found the answer for this important question, which is potentially can be diagnostic marker and a lot of other things. Going to the next question. So no biological answers anymore without network analysis. So again, what we do, we ask the question about microbiota. The second question was, can we pinpoint potential microbe that plays a role or contributes importantly to the development of this syndrome? And what we do, we study a human and we study microbiota. And here we identify 1,800 genes that are specific genes that are up and down regulated. And this is a genes that are specific for enteropathy. As you see, no enteropathy CVAD patients and healthy subjects are very similar between each other, where enteropathy is different here. So, and we also found about 500 different OTUs. OTU is not important, 500 potentially different microbes living in biopsy. So what we do is we integrate this data into the transkingdom network. A transkingdom network is a gene expression with microbial network. And here you see in red upregulated genes and blue downregulated genes, and it's all human genes. And these brown guys are microbes that are enriched in CVAD enteropathy, and greens that are decreased. So I'm not going to go into computational details, but what we identified is that the potentially most important bacteria that would be causing the driving whole gene expression profile of CVAD enteropathy is Acinetobacter baumannii. This was a huge surprise for me because I am ignorant and I didn't know, never heard about this. And some people I still don't know, and some people like my dean said to me, of course I know this bacteria, everybody knows it. So not everybody, but, and I know why he knows, actually. This is actually commensal opportunistic pathogen, and so we can find it elsewhere. But this elsewhere bug can cause hospital-acquired pulmonary blood infection and intensive care units in immunocompromised patients. Never found before in the gut as a cause of disease. But why my dean knows very well about this bug is, try to guess. Nobody? So because this is one of 6 most antibiotic-resistant bugs ever on the list of NIH. And this is extremely important because we know, we might understand now why those patients don't respond to antibiotics. They wouldn't respond to antibiotics because antibiotics wouldn't kill this bug, or there is one antibiotic that kills this bug, not all of them, but it's usually not used. It's all antibiotic from 50s or 60s, colistin. So what we looked, can we test somehow whether this particular bug is actually cause? So because I cannot infect humans, so what we can do in human system, we take a cell line and we do the co-incubation between cell line and the bug and see whether this bug would induce expression of genes that we see in our disease. And this is what happened, and this is a big proportion of pro-inflammatory genes is actually induced in macrophage-like cells by Acinetobacter baumannii. Okay, so the last question about CVAD. What is the last question about CVAD? Why we have a problem with fat absorption? And when we look at these genes, remember this profile of 1,800 genes, what we found is down-regulated genes that is basically tells you problem with absorption, lipid homeostasis down, fatty acid metabolism down, cholesterol transport down, regulation of lipid metabolism, cholesterol transmembrane transport. So this is indication of what we already knew happens in patients. What about upregulated genes? Upregulated genes are mostly immune genes, and the key major feature, common feature of those, a big proportion of this is driven by interference. In our previous study, when we worked with mice, we had some indications that those interference can be actually inducing the problem, although we didn't have good evidence for this. So now we decided to test it. And again, how I can test it? I'm not going to inject in the human being those interference. So what I'm going to do is I'm going to take a cell line, and I don't take cell lines. Obviously, Dasha is taking cell lines. And incubate those cell lines with interferons. So we have evidence that is high production interferon gamma, interferon type 1 also in those diseases, and a lot of genes that in this disease. And we also have clear genes that are responding. So let's look what happens when we treat a cell line, epithelial cell line from duodenum with interference. About 25% of our gene signature is actually can be mimicked just by 2 interferons. And when we do it separately, we have less efficient answer. But what is important is that big proportion of those upregulated genes. So each dot here is gene, and you have on Y, you have upregulation in CVAD enteropathy, and on X, you have upregulation by interference. So all these dots here are genes that are upregulated. This is important. This is immune genes that are responding to interferon. What about these genes? So these genes are very important. These are mostly metabolic genes, and some of them are actually well-known lipid transporters. So, okay, we have gene expression. Who believes in gene expression? So we decided to test it another way, and the test is basically what we do. We are treating cells with interference, and we look about what is changes in cholesterol level in those cells. And what we find, which is very important here, the cholesterol also goes down, and we actually found the cholesterol also goes down if you actually have cholesterol outside in media. And if you don't have media in your well, you have media, but you don't have cholesterol, or you don't have actually serum, which is source of cholesterol, then it will not be affected. So this is a good phenotype in vitro, supporting very well our gene expression analysis. So what else we can do? So we develop other type of analysis that allows us to ask the question using Z networks that you've seen. Which genes are actually downstream of interferon gamma, immune genes that are critical for regulation of metabolism? And when we ask this question, we have our predictions, and when we test them. And the result of the test, and we do siRNA, so we knock down one gene separately, there is nothing. We knock down another gene separately, there is nothing. But when we knock down both genes, we have basically restoration of cholesterol levels, so which means that we found part of the mechanism how interferon control cholesterol levels and lipid transport in those cells. So for now, and it's new results, and it's on working, and we didn't finish yet entirely this everything, although we are writing paper. We have a working model of this disease, and the working model is the following. So it's the major problem here in epithelial cells. As I told you, fat absorption is a major problem, and not just fat absorption, but fat absorption is a major problem here. How it happens? So only IgA is critical player, and potentially one of the most important microbial players is Acinetobacter baumannii, which induce inflammation, and as a result of this inflammation, we have production of interferon gamma and different interferon type 1s. As a result, we have induction of interferon-induced genes that would lead to decrease in fat absorption. Anything you want to know about CVID enteropathy before I jump to the next? No? Okay. So the last one. So I was thinking about going to talk, but because I wanted to show you more of new unpublished data. So this is actually the paper that is not published yet. It's because it's not online, but it's proof of this paper. It was very successful collaboration, again, with Alexander and Richard here, and a lot of other people, as usual in our group. And by the way, I'm not going to tell you anything about diabetes type 2, although this paper is major thing about type 2. Why I'm not going to? It's a big paper. It's a lot of things to talk about. I choose a piece of this paper to demonstrate to you the power of network analysis to answer specific questions. So here we are. We have a question about interferon gamma and Akkermansia muciniphala, and our question would be how interferon gamma can control Akkermansia muciniphala. And can we find this mechanism? Because other part of the paper that hopefully will be soon out, you can read about it. So just for you to believe me that actually interferon gamma is important for controlling Akkermansia muciniphala, I'm showing you two experiments. This one experiment where we have interferon gamma knockout mice, Akkermansia is higher than in wild-type mice. And there is another experiment when we don't use interferon gamma knockout mice. We just block interferon gamma with antibody, and we also see increase in Akkermansia muciniphala. So, okay, Akkermansia muciniphala is controlled by interferon gamma. So what about how it happens? This is our major question. Guys, interferon gamma is a central cytokine in immunity. It's like one of the first things ever discovered by immunologists. We know so many things. And if I start testing what is actually important here, we can take all this room, put in the lab, and we will never finish in the next 10 years. So this is why we need networks, and this is why we need system biology approaches. So what we do first, we are looking which genes are in the mice regulated by interferon gamma, and more than it, we need to do it a little bit smarter. We need to know which genes are regulated by interferon gamma and not dependent on Akkermansia, because those that are regulated by Akkermansia, they're afterwards, right? So this is why we are comparing wild-type mice with interferon gamma knockouts negative and positive, and those would be common for this comparison. This is the genes. Among those, we are going to look and investigate which ones are critical. So here we found about 200 genes that would be regulated into interferon gamma knockout mice the same way whether you have or don't have Akkermansia mucinifera, differently from wild-type mice. And the questions now, can we look into those 200, it's a little bit more than 200 genes, and find which of those are important? So to answer these questions, we go and do whatever we're doing usually. We reconstruct gene expression network. Here's the gene expression network of interferon-dependent genes. And we connect this interferon-dependent network with Akkermansia mucinifera. And then we rank all our interferon-dependent genes by certain criteria where connection and location in that network is important. And now we have a list of our candidates that are critical in the controlling Akkermansia mucinifera under regulation by interferon gamma. So all these genes are regulated by interferon gamma. Some of those may be critical. So what we do, we find collaborators that have IRGM1 knockout mouse. And those collaborators actually were doing the experiments. And our prediction is if it's IRGM1 knockout mouse, so as in interferon gamma knockout mice, Akkermansia mucinifera should be higher than in the controls. Here we go. We have two different facilities where they keep their mice. And in both facilities, you see that IgM knockout mice have higher amount of Akkermansia mucinifera than in wild type. Interestingly enough, the levels, overall level of Akkermansia is very different in two facilities, which is not exactly surprising because this is specific pathogen-free. This is a so-called dirty facility, conventional mice. So level of Akkermansia is much lower, and we know that it can be competed by other bugs. What is important, the difference are still the same. So we have increased levels in IgM1 knockouts, which is supporting our prediction. So we did answer. So this is a way how, and it's okay, by guessing, by reading the literature, we would never ever would be able to figure out how interferon gamma controls Akkermansia mucinifera. Only network analysis would help us. So this is the end. What I want to tell you today is that our group is doing our research about biological questions, medical questions, and what we do a lot, we study human and mice, and we collect a lot of big data. When we collect about big data, it's about host and, of course, about microbiome, because we studied host microbiota interaction. And then comes very important step, the network analysis, which allows us to pinpoint our candidates, whether it would be microbial genes, I didn't have time to show this, microbes themselves, or host genes that we test afterwards in experiments, whether it's in vitro experiments or germ-free facility. We have wonderful germ-free facility in this campus since at least we came here. So the only thing that I wanted to say here, so I tried to mention all our collaborators, but so the major person, as you can imagine, according to Mark, and he is right and I am honest, is of course Dr. Shulzhenko, and she decides everything. When basically when we are right, she is what she's what is what. If something goes wrong, it's probably my fault. She decides what I should wear today. So, and I mean, I've been lucky, and I guess. So there is one important person in the story, though, besides Dr. Shulzhenko, which is Dr. Yambartsev, who was visiting this college 2 times by now for sabbatical. And he was the person who convinced me back then that we should start working with networks, not just compare gene expression, compare, correlate, and whatever. We need to use networks. And he was essential to start, and he was involved in a lot of development, and we have other collaborators now because of him in mathematical department here, Professor Kovchegov. So, and of course, when I said I, if I mention ever I, it's of course we, and when I said we, it's basically they, and they are, so we have two wonderful people at the beginning in our lab, two postdocs. One is our computational genius who unfortunately is leaving me now, which is, I'm very happy for him, by the way. He is going to continue as a postdoctoral or research associate in Harvard Medical School before he gets his own lab. And Renee, she is more biologist. And of course, we have Stephanie, who is genius with our germ-free facility. And Kim, I mentioned already. Where is Kim? No, I don't see here, but he was working with microbiome and cancer. And Jalu used to be our postdoc for a short time. Very good guy, but didn't work very well. Manoj is actually, it didn't work between us. It's not that he didn't work. Okay, I'm sorry. He's too computational. He was not so much interested in biology. I was telling to computational scientists, you came here to treat cervical cancer. He didn't understand me. So anyway, Brianna is an undergraduate student, and Manoj was doing the THP1 experiments, and Richard is our new computational postdoc, our future star. You will see his data for future. You will be all very happy. And of course, okay, Oregon State, who is funding everything, and in particular College of Pharmacy and College of Veterinary Medicine contribute some to this. Of course, NIH is funding our research right now, and Medical Research Foundation a little bit. And we have some funding from NSF for developing our computational new approaches. And I'm a little bit out of time, but I want to thank you all for coming here today. Since you said that you're... Okay, Mark said that he's leaving.
I have a question on cervical cancer.
Yes.
So can I ask now or?
Yeah. No. Because more people will be helping me.
So progression of the cancer, like in all cancers. So what is, so you're expecting is going to be a different interaction now between microbiome and host and microbiome. And these interactions are going to be different as the cancer progresses.
Yes.
So your studies, I guess, were on the remnant phase. So what did you expect? What is your prediction from the initiation or from the viral infection to the
This is right to the point. This is a major thing, I guess, in the whole cervical cancer story. So at the beginning, you have microbes there that would probably contribute potentially, it's not necessarily the same microbes, maybe different, to increase susceptibility and chronicity of viral infection, which is requirement for developing of the cancer. However, the bacteria is still there, and it can be very different bacteria and very different mechanism how bacteria contributes to elimination of the virus, because at the end it has to be eliminated. So it is very important questions. Unfortunately, at this point we don't have early stage data at all and early stage samples. So we are working with, at this point, with different stages of invasive cancer that might shed some light to this question. But the true correct answers would be coming if we would get enough samples from pre-cancerous lesions. And I do expect would be different bacteria contributing in each step. Did I answer?
So
HPV?
and not to something else. So I guess
Oh
that's
yeah,
actually
HPV. So it's quite simple at this point. No cervical cancer happens without HPV. Most of people who are infected by HPV never get cervical cancer.
no,
No?
it's a different question. So the interaction that you see between host and microbiome in cervical cancer, is this unique to the cervical cancer?
We are at preliminary stage. This is also a very good question actually, because some bacteria that we find, and we don't know yet, because our testing experiments are not exactly at the stage that I would like them to be. Some bacteria are actually also found increase in bacterial vaginosis, and bacterial vaginosis was found to be associated with cervical cancer. So the bacteria that we find as a potential candidates contributing to cervical cancer were shown to be associated with bacterial vaginosis. How bacterial vaginosis interacts with cervical cancer, we don't know, but we know by epidemiology that there is an interaction.
And one more question, if I can, on your, when you tried to answer. So did you say that the HPV is specific for the cervical, so causes cervical cancer,
Yes.
but does not cause any other type of cancer?
No. HPV can cause oropharyngeal cancer, anal cancer, penile cancer, and this is actually a very good question, because when I came here 5 years ago and I get the same answer, I gave the wrong answer. Sorry. I said it's just cervical cancer. I don't know what I was thinking. So, and I remember it until now because it's a shame. But I know the right answer now. Anal cancer, penile cancer, oropharyngeal cancer. In fact, for oropharyngeal cancer, we have emerging epidemic of oropharyngeal cancer, HPV associated, not tobacco, not other reasons.
And that now leads to my final question. So if you compare now the network of the HPV-dependent oropharyngeal cancer with the cervical cancer, did you try to say the same?
So, very good question. Unfortunately, so basically I was doing some analysis some time ago where comparing cervical cancer with oropharyngeal cancer, and there are certain group of key drivers that are the same, at least the same way regulated in oropharyngeal cancer and in cervical cancer. However, it didn't go that far, mostly for unavailability of pursue all interesting questions. Yes, of course.
So you have published about LAMP3 already about regulating through that as a true copy number variation by suppressing clearance of invalid patients. But you mentioned that there is also another network which is the cell cycle. Are there genes that are critical for that process?
So, among cell cycle genes, we don't see genes that would be actually regulating potentially antiviral response, but they're critical for cell growth. So for sure we will know more hopefully soon, because now we are transitioning from one technology to another. I didn't want to talk about this today because I don't have the results yet, but hopefully we will drop the price of RNA-seq about 5 times or more in the very next future. Dasha already prepared the libraries and that CGRB, and so we will be able to test more things, and we are planning to answer this question. The only thing that I know for sure is that the results, Dasha's results on key drivers, that they inhibit, most of them inhibit the cell growth, different cell lines, by the way, cervical cancers. This is very consistent result, and done not like was repeated recently just to test new media in the lab. We were testing changing to cheaper media, just testing whether siRNA would give the same result.
And maybe the last question is, in your second part of the story, you talked about the interferon effects on CDID gene expressions. Are they type 1 interferons? Or
Both.
both?
Yeah, so we did test, there was no time to talk about this. We did test separately. The impression is that it's actually interferon gamma has more stronger effect, but it is, I wouldn't be too about this. So we definitely have both interferons in disease, therefore we are testing both. So some of the, probably type 1 is contributing less, but it's dose-dependent issue when you work with cell line. Yep.
So in the case of CDID, I have two questions. One, did you see the effect on interferon gamma with other bacteria, like Clostridium or E. coli or Enterobacter? Did you test a range of bacteria and see if you see the same kind of gene upregulation dynamics that you see with acid?
So Acinetobacter is actually controlled by IgA, not by interferon gamma.
Sorry, then I got it backwards, yeah.
So, and there are several, so out of 500 something that you use, we have about 70 that is potentially controlled by IgA. Among those potentially controlled by IgA, about 36 of them were increased in enteropathy. So among those, Acinetobacter were the best candidate to be regulating this. There is another very abundant microbe. The most similar, what we think it is Lactobacillus plantarum, that we believe is not supposed to induce the same effect, and the experiment is ongoing. The control experiment with Lactobacillus also increased in enteropathy, but we don't predict it as being inducing this gene expression profile.
Great. And then my second question comes from like the percentage of enteropathy and CDID. Do you see like, is there a higher incidence in say the Middle East versus the US versus?
The quality of this epidemiological data on this, not that great. So the range of different reports is from 10 to 35, 40% in different studies. And I believe that the reason is because there are a little bit different diagnostic criteria. The last very recent study was, I don't even remember what they said. But basically, there is no obvious connection that I could know. But maybe there is. Joshi, do you remember anything about the last question? No? Anything about Middle East and enteropathy?
Yeah, the reason I ask is that Acinetobacter is a huge problem, and incremental in the Middle East is very prevalent. So it could possibly be a higher incidence when you find more of them in theory in Middle Eastern dwelling.
So this particular patient population is usually very bad patients. They are treated with multiple antibiotics. They are treated with corticoids. This is like total mass patients, unfortunately, and they obviously receive global immunoglobulin, which is basically IgG. So the reason for Acinetobacter in those guys, I do believe, is related to their multiple exposure to different type of hospitals before they even ever diagnosed with CVID at all, rather than anything else, because Acinetobacter is there quite a lot. And in fact, there was paper about NIH clinical center with Acinetobacter there, which those people are not NIH living patients. They actually come only, so it's not, I don't think it's from NIH. They caught this Acinetobacter baumannii. And another thing I didn't have time to mention, So there were reports, and we also detect this, is norovirus can be contributing factor. We're actually testing now in animal model the contribution of Acinetobacter and norovirus together. There is no human model for norovirus in vitro, I mean, yet.